Hello and welcome to the thin layer chromatography experiment for the CBCH lab. The objective of this lab is to visualize the extracted lipids of our sample. Thin layer chromatography, or TLC, is a technique used to separate non-volatile mixtures. Thin layer chromatography is performed on a sheet of inert substrate such as glass, plastic, or aluminum foil, which is coated with a thin layer of adsorbent material, usually silica gel, aluminum oxide, or cellulose. This thin layer of absorbent material is known as the stationary phase. After the sample has been applied to the plate, a solvent or solvent mixture known as the mobile phase is drawn up the plate via capillary action. Because different materials ascend the TLC plate at different rates, separation of the sample is achieved. To begin this experiment, 5 mL of the mobile phase was made. Only enough mobile phase is needed to submerge the bottom of the plate and the paper filters on the side of the chamber. The mobile phase consisted of 3.4 mL of isopropanol, 227 microliters of ethyl acetate, 227 microliters of ammonium hydroxide, and 1.14 mL of water. These components were mixed together in a glass graduated cylinder to prevent plastic contamination. The mobile phase was added to the glass TLC chamber. Paper filters were placed in the chamber and also covered in the mobile phase to allow for the vapors to fill the entire chamber. The dried sample from last lab was resuspended in a 1:1 chloroform methanol solution. – 20 microliters of the sample was added to the silica TLC plate. The plate was placed in the glass TLC chamber. The chamber was sealed with parafilm to prevent any vapors from escaping. We then waited for the mobile phase to reach the top of the plate, which took about 25 minutes. The plate was then removed from the chamber and allowed to air dry for about 5 minutes. The plate was then placed in a chamber containing iodine crystals to allow for the visualization of the bands or spots on the plate. Iodine is one of the oldest visualization methods used. It works because iodine has a high affinity for both unsaturated and aromatic compounds. It will bind to these compounds and turn them into a dark brown color. We waited for the plate to turn light brown and for the dark brown bands to appear, which took about 45 minutes. Here is a time lapse of the process. After removing the plate from the chamber, the bands are visible and a ruler can then be used to measure the retardation factor or the retention factor, which is the distance traveled by the sample divided by the total distance traveled by the mobile phase. Lipids can have unique retention factors, which can aid in their identification.